In the last class, we completed the blood groups and sex determination up to certain extent. In this class, we are going to complete the remaining part of sex determination, sex-linked inheritance, and gene expression regulation up to animal breeding. We are going to discuss what type of questions are going to asked in the EAMCET examination. As usual, while discussing each and every question, related each question, we have to discuss some important bits. Now we go for the remaining part of blood groups and sex determination. First we have to observe the question: how many genotypes are possible with 3 alleles? You observe the question: how many genotypes are possible with 3 alleles? Now regarding the blood groups, according to Bernstein, this one we call it as IA, and this is IB, and this is IO. Now 1, 2, 3 alleles are present. IA gives rise to A antigen, IB gives rise to B antigen, IO does not give any antigen. Here you have to observe how many alleles? 3 alleles. Now here what is the gene I? The gene I has more than 2 alleles, IA, IB, IO. In order to get the multiple alleles, we are having a formula n plus 1 by 2. Instead of n, we use 3 plus 1 by 2. 4 into 3, 12 by 2. We are getting how many genotypes? 6 genotypes. Already we know very well how many blood groups phenotypically? 4. A blood group, B blood group, AB and O. Phenotypically 4. Now the A blood group has IA, IO, 2 genotypes. B blood group has IB, IO, 2 genotypes. AB blood group, it has 1 genotype, IB. What about the O blood group? IO. Total number of genotypes 6. Now how many genotypes are possible with 3 alleles? 6 genotypes. Now the next question: which blood group does not appear in the progeny if one of the parents is AB group? Now again you observe the question: which blood group does not appear in the progeny if one of the parents is IB? For example, one parent blood group is AB, IA, IB. Now you have to observe the first one, A group. Now IA. Now if we go for the cross, we are getting A blood group, and next AB blood group and AB blood group. Now the AB is crossed with A, we are getting these blood groups. Now the second one is B. Now this is IB, and this is IA, IB. Now if we go for the cross, we are getting AB, and next B blood group, and we are getting the B blood group, and again we are getting the B blood groups. Now the third one, now again the parent is IA, IB, and next what is the third option? AB, that is IA, IB. Now if we go for the cross, we are getting A blood group, and next AB blood group, and next AB blood group, and next B blood group. Now as we observe the question, which blood group does not appear in the progeny if one of the parents is AB group? The answer is we should not get the O blood group. Now the answer is fourth one. Now the next question. Which blood group is not possible in the children if one of the parents is O group? Now you observe the question. Which blood group is not possible in the children if one of the parents is O group? Now we know very well the O group, IO, IO. Now if it is crossed, A group, now IA, IA. Now we are getting A, and A. Now the second one, B, IB, IB. One of the parents is IO. Now we are getting the B, and B. Now the third option is IA, IB, and IO, IO. We are getting A, B, A, and B. Now again observe the question. Which blood group is not possible in the children if one of the parents is O group? We are not getting AB blood group. Anywhere we are not getting the AB blood group. Now the answer is third one. Next question. Sex determination in most of the organisms is by. Now as we observe the sex determination, now we go for the body characters. CHT means body characters. The genes responsible for the body characters are present on autosomes. Now the genes responsible for the sex are present on what type of chromosomes? Allosomes. What is the other name of allosomes? Sex chromosomes. Now here the question is sex determination in most of the organisms is by allosomes. Here the answer is allosomes. What about the autosomes? On autosomes the genes for body characters are present. What about the hormones? For the sex differentiation, what are required hormones? But here sex determination is by allosomes or sex chromosomes. Now next question is sex of the offspring depends on fertilizing the sperm in case of. Now as we observe the sex determination, one method is XX, X0. Now this is the female, this is the male. Now here you have to observe, here the sex chromosomes 2 in number in female, but in male how many sex chromosomes? Only 1. Second sex chromosome absent indicated by 0. It means in this method the male gives 2 kinds of sperms. In one sperm X chromosome present, in one sperm there is no sex chromosome. Now this one gives the 2 ova, each ovum has the X chromosome. Now the X bearing sperms fertilizes with this ovum, you are getting the female. Again this sperm fertilizes with this ovum, you are getting the female. Now here the sperm has no sex chromosome, this sperm fertilizes with this ovum, we are getting the male. This sperm fertilizes with this ovum, we are getting the male. So students, as we observe this method, in this method XX female, X0 male, here the male produces 2 kinds of sperms. In this method sex determination based on type of sperm fertilizes, because here the male is heterogametic, it produces two kinds of gametes, where we have to observe this type of method. One example is Hemiptera, that means bed bugs, and the second one we call it as Arthroptera, example is cockroach, and the third one we call it as grasshopper. Now here, what is the answer for this question is grasshopper. In grasshopper, what is the method XX, X0? The male heterogametic, it gives two kinds of sperms. So sex determination based on type of sperm fertilizes. Next question. Failure of the paired chromosomes to segregate during anaphase of meiosis is called. Now again you observe the question, failure of paired chromosomes. For example, take the human being body. In the human being body, female and male. Now in female, autosomes, sex chromosomes. Male, autosomes, sex chromosomes. Now during the formation of gametes, the chromosomes equally separated. If the chromosomes are equally separated, you are getting AX. AX. Here you are getting AX. You are getting AY. Sometimes at the time of formation of gametes, the sex chromosomes are not separatedly in a correct order. Then we are getting, for example, you observe the female. In female, XX. If the chromosomes are not separated separately, or if they are not separated in a correct order, we are getting AAX, and next AAXX, or we are getting AAX, and we are getting AXX. So here the chromosomes are not separated in a proper way. If the chromosomes are not separated in a proper way, that is called non-disjunction. Because of the non-disjunction, that means chromosomes are not separated properly, we are getting abnormal gametes. If abnormal gametes undergo fertilization, we are getting the variety of male and the female individuals. Here the question is the failure of the paired chromosomes to segregate during anaphase of meiosis is called, they failed to separate. That means they are unable to separate. That we call it as non-disjunction. The answer is the non-disjunction of chromosomes. Next question. In human beings, the monozomy of 23rd pair chromosome causes. Now what is meant by monozomy? That is 2n-1. From the diploid chromosomes, we remove one chromosome. For example, this is first pair of chromosomes, second pair, third pair, fourth, fifth, sixth, seventh, eighth, ninth, 10th. 11th pair, 12th, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. So 22 pairs autosomes, and this one we call it as, for example, if this is female, the 23rd pair is XX. If it is the male, the 23rd pair is XY. Here what they are asking, in human beings, monosomy of 23rd pair. Monosomy means from the diploid chromosome, one chromosome is removed. Here this is 23rd pair. In the 23rd pair, XX chromosomes are present. Now you remove one chromosome. Now how many autosomes? 44. How many sex chromosomes? 1. 44 plus 1, 45. Now this is a female human being suffering from Turner's syndrome. Now here, because of the monosomy, the female human being suffering from what type of disease? The answer is the Turner syndrome. Now we go for the next question. Now we have to discuss the sex-linked inheritance up to the animal breeding. Now what is the first question? A red-eyed male. You observe the question. Red-eyed male. Red-eyed male and red-eyed female. White-eyed male, white-eyed female. Now here I am displaying the red-eyed female, red-eyed male, white-eyed male, white-eyed female. 1, 1, 1, 1. Now red-eyed male and female, white-eyed male and white-eyed female. Drosophillas appear in the progeny when a cross is made between. Now this is the male. The male is white-eyed. On X chromosome smaller. Now we take the female, red-eyed but heterozygous, capital R, small r. The heterozygous is also called carrier. Now we call it as carrier or heterozygous. Now we go for the cross. We are getting XX, capital R, small r. XX, female, capital R, small r, red. Now these two are crossed. XX, small r, female. We are getting white-eyed. Now this X crossed with Y, capital R, XY, male. We are getting the red. Now XY crossed, small r, male, white-eyed. Now you observe, whenever white-eyed male crossed with carrier or heterozygous red-eyed female, you are getting the children in 1:1:1:1. Now you have to observe one red-eyed female, red-eyed male, white-eyed male, white-eyed female. Now the answer is white-eyed male crossed with the red-eyed carrier female. Now the next question. Assertion. Duchenne muscular dystrophy is more common in males than females. Now Duchenne muscular dystrophy, this is the short word DMD. Duchenne muscular dystrophy. It is a disease. The gene responsible for this disease is recessive. It is present on X, on which chromosome? X chromosome. Now, as we observe the Duchenne muscular dystrophy, it is a genetical disorder. The gene for this disease is recessive, present on X chromosome. The one who has been suffering from this disease in his body from the birth onwards, slowly all muscle cells undergo contraction, there is no relaxation. So slowly whenever the patient enters into 16th year or 17th year or 18th year, definitely the patient must die, because in his body all muscles are contracted, there is no relaxation. Now this DMD, Duchenne muscular dystrophy, is more common in male, because as we observe the male animal, XY chromosome present. This disease controlled by a gene that is recessive, that recessive gene present on X chromosome. So in male how many X chromosomes are present? 1. So the disease is more common in male. In order to express in female, in order to express that recessive gene must be present on both X chromosomes. So the DMD is more in males, because the males having how many X chromosomes? 1. So assertion is correct. And reason, here reason, the males are hemizygous for the Hollandric genes. Now as we observe the male parent, in the male parent this is X chromosome and this is Y chromosome. X and Y chromosomes are separate, they are called non-homologous. The genes present on X chromosome, they are haploid N. The genes present on Y chromosome, haploid. The haploid is also called hemizygous. The haploid genes present on Y chromosome are called technically Hollandric genes. Now here you have to observe assertion. Duchenne muscular dystrophy is more common in males than females. So assertion is correct. What about the reason? The males are hemizygous. Hemizygous means haploid for Hollandric genes. What are the Hollandric genes? They are present on Y chromosome. So A and R are correct. R does not explain the A. So both are correct. R does not explain A. Next question. Hemophilia B. Now we are asking the hemophilia B. As we observe the hemophilia, it is a genetical disorder. It is popularly known as Bladder's disease. The one who has been suffering from the hemophilia, if the person gets injury, there is a continuous loss of blood. Today or tomorrow or day after tomorrow, the hemophilic patient is going to die. It is also known as the Bladder's disease or royal family disease. The hemophilia is classified into hemophilia A, hemophilia B, hemophilia C. The gene for hemophilia C present on autosomes, don't forget it. The genes for hemophilia A, hemophilia B present on allosomes. Okay. The one who has been suffering from the hemophilia A, in his body the 8th factor is not produced. The one who has been suffering from the hemophilia B, in his body the 9th factor does not produce. The one who has been suffering from the hemophilia C, in his body 11th factor does not produce. We know very well, for the coagulation of blood how many factors are responsible? 13. But according to the recent investigation, the 6th factor is removed. So anyhow, for the blood coagulation 13 factors are required. If 13 factors play an important role, the blood will coagulate within 3 to 6 minutes. If not, the blood continuously oozes. that is a genetical disorder that is called Bladder's disease or royal family disease, because in the universe the royal family word is applicable to only British family. In the British family, one generation suffered from the hemophilia, so it is called the royal family disease. So here what we are asking, hemophilia B is due to the deficiency of the ninth factor. So what is the name of the ninth factor? Plasma thromboplastin component. The answer is, now we go for the next question. When a woman heterozygous for incontinentia pigmenti, what is meant by incontinentia pigmenti? As we observe our skin has some coloration due to the presence of melanin. The one who has been suffering from the incontinentia pigmenti, in his body the melanin pigment removes randomly. Where melanin removes, there we are getting the white patches. Don't call it as a bully disease. It is incontinentia pigmenti. Because of the incontinentia pigmenti, from the body randomly the melanin pigment losses. Where melanin pigment absent, there we are getting the white patches. The gene responsible for this disease is dominant. This dominant gene present on X chromosome. So the gene for the incontinentia pigmenti is present on X chromosome. It is dominant. Here what we are asking, when a woman heterozygous for incontinentia pigmenti. Now the female suffering from incontinentia pigmenti. What is the genotype? Capital I, small I. Because here we are asking a woman heterozygous for incontinentia pigmenti. Now marries a normal man. The man has no incontinentia pigmenti, normal. Small I. Now we go for the cross. Capital X, capital X. Capital I, small I, XX, female. Now this female has incontinentia pigmenti. Okay. Now we go for this cross. Small I, small I. This is female, but no incontinentia pigmenti. N means no incontinentia pigmenti. Now we go for this cross, XY, capital I, XY, male. Now suffering from incontinentia pigmenti. Now this one, XY, small y. Now XY, so male. N means no incontinentia pigmenti. Now as we observe, one daughter has disease, one daughter has no. One son has disease, one son has no disease. Now here what is the answer? Half of the progeny means children, half of the children will have incontinentia pigmenti. Now here incontinentia pigmenti. Out of 4, 2 suffering from incontinentia pigmenti. So half of the children getting the disease. Now the next question. In an experiment, male Drosophila, here male Drosophila with bobbed bristles. What is meant by bobbed bristles? Bobbed bristles are soft. What is meant by Drosophila? It is a type of fly, commonly known as fruit fly. Scientific name is Drosophila melanogaster. Here we are asking male Drosophila with bobbed bristles mated with a homozygous wild female. What is meant by wild? The bristles are normal. What is the meaning of bristle? Hairs. What is meant by wild bristles? Normal hairs. What is meant by bobbed bristles? Short hairs. Now we go for the cross. Now here the male Drosophila with bobbed bristles. Now this is the male Drosophila with bobbed bristles. Now the bobbed bristles controlled by a recessive gene present on both small b, crossed with the female. The female is said to be wild. The male is said to be bobbed. Now this is the genotype. Now the female is wild, that means plus plus. Okay, we go for the cross. Now XX small b plus XX small b plus, here small b plus, this is female, this is female, small b plus, no bobbed bridges, wild, this is also wild. Now we go for this cross. Now here plus b, male, no bobbed bridges, wild. We go for the next cross, plus b, male, wild. Now you observe F1 generation. Whenever the bobbed male crossed with wild female, you observe the cross. In the F1 generation, we are getting all wild. That means the bridges are normal. Now you take any two individuals and go for the F2 generation. You use checkerboard. Now the male and the female. Now here X, B, X plus. What about the male? We take X plus Y, B. You observe the cross. Now X, B, X plus, X plus, X plus, X, Y, B, B, and X, Y plus B. Now here 2X. Now no doubt this is female. This is female. XY, male. XY, male. Now you observe the genotype B and plus. It is wild. Now B plus, it is wild. And this is 2 plus, wild. And this is small b, bobbed. Now in the F2 generation, we are getting the 4 offsprings. Out of 4 offsprings, how many are wild? 3. How many bobbed bridges? 1. Now we are asking, you observe the question, in an experiment, male Drosophila with bobbed bridges mated with homozygous wild female Drosophila. Please stop. In the F2 progeny, wild condition and bobbed bridges appear in a ratio of 3:1. Now the answer is, next question. In the experiment conducted by Avery, McLeod, M. Carty, in the experiments conducted by Avery, McLeod, Carty, transformation did not occur when S filtrate was treated with. Now, as we observe the transformation experiments, the transformation experiments first conducted by Griffith. Now he conducted experiments on Staphylococcus pneumonia or Diplococcus pneumonia. Here the name of bacterium is Diplococcus pneumonia. In the Diplococcus pneumonia, 2 strains are present. One is R strain and one is S strain. Now the Diplococcus pneumonia, why it is called Diplococcus? 2 strains are present. Diplo, the shape of the strain is round, so coccus. It causes pneumonia fever, so called pneumonia. The Griffith conducted transformation experiments on Diplococcus pneumonia. After that, the transformation experiments continued by Avery, McLeod and Carty. Now they treated the material of S with protease enzyme and with lipase enzyme and with amylase enzyme and treated with DNAs whenever the material of S treated with protease enzyme, there is transformation. Whenever the material treated with lipase, there is transformation. Whenever the material treated with amylase, there is transformation. Whenever the material treated with DNase, there is no transformation, because the DNase enzyme digest what type of material? DNA. So in the S strain, what type of material is present? DNA. The DNA material digested by what type of enzyme? DNase. So whenever the material of S is treated with DNase, there is no transformation. Whenever it is treated with the protease, lipase, amylase, the material in the S strain is not digested, so it is transferred. Now here what they are asking, in the experiments conducted by Avery, McLeod and Carty, transformation did not occur when the S filtrate was treated with what type of enzyme? The DNase. Next question. Central dogma of molecular biology is the flow of genetic information. Now as we observe the central dogma of molecular biology explained by Watson and Crick. Now as we observe the central dogma of molecular biology, this is the DNA. Now in the DNA two strands are present, 5', 3', 3' and 5'. Now at the time of synthesis of mRNA, which strand acts as a template? 3', 5'. Now the DNA gives rise to mRNA. The mRNA synthesized in which direction? 5', 3', because it is complementary to this strand. Now from the template mRNA formed by a process called TR, transcription. From the mRNA by a process called translation, we are getting the polypeptide chain. Polypeptide chain. That polypeptide chain becomes the protein. Now what is meant by central dogma of molecular biology? In each cell, the DNA transforms the biological information to the protein through mRNA. Now this protein helps to produce a structure. The protein helps to give a proper shape. The protein helps to perform a function. It means in our body or in the body of living organisms, in each cell, the DNA transforms its biological information through mRNA to the protein. With the help of that protein, in our body, every cell performs its activity. That is called central dogma of molecular biology. Now here you have to observe, what is the central dogma? DNA gives rise to mRNA, that gives rise to the protein. Now the correct answer is DNA, RNA means here mRNA and protein. The next one. In the lac operon of E. coli, enzyme specified by Z gene causes. Now as we observe the lac operon concept, now this is the DNA strand. Okay, this is DNA strand. Now in the prokaryotes, as we observe the gene expression and regulation, how many structural genes are present? A, Y, Z. On the left side of A, what is present? Y. On the left side of Y, what is present? Z. On the right side of Z, Y. On the right side of Y, what is present? A. Now this gene gives rise to what type of enzyme? Transacetylase. Its function is not known till the minute. Why? This gene gives rise to permease. This gene gives rise to an enzyme called permease. Now what is the function of permease? With the help of permease, the E. coli bacterium absorbs the lactose. Now what about the Z gene? It synthesizes beta galactosidase. Now what is the function of beta galactosidase? It converts the lactose into glucose and galactose. Now here what is the question? In the lac operon concept of E. coli, the enzyme specified by Z gene. What is the enzyme specified by Z gene? Beta galactosidase. Now it causes hydrolysis of lactose. That means the lactose hydrolyzed into glucose and galactose. So the answer is, now the next question, split genes of eukaryotes having split genes of eukaryotes. Now we are discussing the eukaryotes. What are the eukaryotes? Distinct nucleus with nuclear membrane present. What are the eukaryotes? One example is protista, and one example is fungi, one example is plantae, one example is animalia. Now all these are the eukaryotes. Now in eukaryotes, as we observe the DNA strand, now this is 3 dash 5 dash. Now here what are present? Exon, intron, exon, intron, exon, intron, exon, intron. E means exon, I means intron. So in eukaryotes, 2 different genes are present, so called split genes. Okay. Now this gives rise to the heterogeneous nuclear RNA. It has exon, intron, exon, intron, exon, intron. Now the exon performs transcription as well as the translation. That means the exon gives rise to mRNA, that mRNA gives rise to protein. Now what about the intron? The intron performs only transcription but not translation. So here in eukaryotes, intron genes are not helpful for coding the proteins. So introns are non-coding, exons are coding. Now in eukaryotes, split genes are, exons are coding for the proteins, introns are non-coding for the proteins. So the answer is 3. Next question. Sickle cell hemoglobin is formed due to the replacement of. Now the sickle cell anemia. Now as we observe the human beings, in the body of human beings, what is the shape of RBC? Biconcave and circular. That is RBC. In the RBC, what is present? Hemoglobin. Now this is B, that is a protein. Now it has alpha gene and beta gene. Now it has alpha 1, alpha 2, 2 polypeptides, and this is beta 1 and beta 2. How many polypeptide chains are present? 4. So it is called tetramer. Now RBC shape is circular and biconcave. Now as we observe the beta gene, in this gene, in the 6th place, what is present? Glutamic acid. Okay, in all human beings body, in the hemoglobin, in the beta gene, at 6th place glutamic acid present, so that the RBC is biconcave and circular. If this glutamic acid is replaced by valine, what is going to happen? The shape of RBC changes, sickle shape. So RBC size decreases, hemoglobin percentage decreases, the person suffers from anemia. After few days the person is going to die. Now here sickle cell hemoglobin is formed due to the replacement of glutamic acid by valine. Now the answer is, next question, the practice of breeding sires of a given breed to a non-descriptive female and their offspring for generation after generation. Now we have to observe the cross. This is one male individual. Now it is considered as ox. Now this is one female individual considered as cow. Now in this male animal we are introducing one character. Okay, CHD means character. In this male animal we are introducing one character. Now this male is called descriptive. Now the female is said to be non-descriptive. Now this male and female are crossed. We are getting a female offspring. Now we want to improve the quality of introducing character. What is going to done? Always the female offspring crossed with descriptive male crossed with non-descriptive, we are getting female offspring crossed with descriptive male, again male crossed with the female, getting the female offspring crossed with the descriptive male. That process we call it as grading up or upgrading. Because of this process, what is going to happen? The introducing character quality after a number of generations increasing. Students, again repeating, this is the male animal. We are introducing one character. Now the male is said to be descriptive. This descriptive male crossed with the non-descriptive female, we are getting the female offspring. Now generation to generation we are crossed the female offspring with the descriptive male. That we call it as grading up or upgrading. So after few generations the quality of introducing character increases. Now you observe the answer. Now the next question. Heterosis means. What is meant by heterosis? Heterosis means the vigor of hybrid. What is meant by vigor? The capacity of reproduction. Now take for example, this is a tall plant. The genotype is capital T, capital D. This is dwarf plant. The genotype is small t, small t. This is homozygous dominant, homozygous recessive. This is one gamete, another gamete. Now we are getting capital T, small t. Now this one we call it as hybrid. Okay. Now what is the genotype? Capital T, small t. when compared to that of parents, this hybrid offspring having more vigor, because whenever capital T crossed with capital T, always we are getting only homozygous at all. In the same way, whenever small t is crossed with small t, always we are getting the homozygous dwarf. But as we observe this offspring, capital T small t is crossed with capital T small t, we are getting capital T, capital T small t, small t. So, the hybrid offspring has more reproductive capacity than that of its parents. Now here, what is the heterosis means? You observe the answer. Increased vigor resulted in the offsprings of unrelated animals cross. This is one parent, another parent. Both are unrelated. We are getting what type of offspring? Capital T small t. That is hybrid. Now this hybrid offspring having more vigor than its parent, because you have to observe the cross. This hybrid on a self-pollination gives capital T, capital T small t and small t. Now we go for the next question. Arrange the correct sequence in the cloning procedure of Dolly. Now, the cloning of organisms first conducted by Ian Wilmot. He was confined to England. Recently, the Dolly died. Now the same person, Ian Wilmot, again conducting the cloning experiments. Now he has to improving the lifespan of the offspring. Now in the cloning experiment, what he did? Now as we observe the cloning experiment, first he has taken one female sheep. From this female sheep, he has taken somatic cell. The somatic cell is also called udder cell. It is also called vegetative cell. Okay. Now this is vegetative cell, this is the nucleus. Now he has taken another female sheep. From this female sheep, he has taken ovum. Now in the ovum nucleus present. Now he removed the nucleus. It is called denucleated. Now in the ovum no nucleus, but in this vegetative cell nucleus present. Now in the laboratory these two are crossed. Now he got the zygote. Now because of the electrical stimulus, it undergoes the cleavages and the zygote becomes the morula. The morula becomes blastocyst. Now this blastocyst is introduced into another female sheep. Now here this female is called surrogate mother. This female is said to be surrogate mother. In Telugu it is called Marudu Talli. The surrogate mother, she carries embryo of other animal. Now in this experiment, he used how many female sheep? 1, 2, 3. First vegetative cell has taken, next ovum, nucleus removed, then these two are crossed zygote, underwent cleavages, obtained morula, blastocyst. It is introduced in the uterus of the third female sheep. Now after completing the experiment, this surrogate mother has given birth to a cloning animal that was named as Dolly by Ian Wilmot. Now the Dolly resembles this female sheep, because from this female sheep he has taken vegetative cell. In this vegetative cell, what is present? The nucleus. So the dolly resembles the female sheep which has given the vegetative cell, but not this female sheep, because from this female sheep has taken the ovum, but from this ovum what is removed? The nucleus is removed. Now based on this, you have to arrange the points in a order. One is blastocyst, isolation, zygote formation, denucleation, surrogate mother. The first one is vegetative cell is isolated. Second, from the ovum nucleus removed, denucleation. Third one, zygote formation. Fourth one, blastocyst formation. Last one, the blastocyst is kept in the uterus of surrogate mother. Next question. Transgenic animals can be produced with the help of transgenic animals can be produced with the help of the transgenic animals first developed by Palmiter and Brinter. Now, Palmiter and Brinter taken the gene responsible for the growth hormone. Now, from the body of human being or from the body of rabbit, they have taken the gene responsible for the growth hormone. Now, this is the gene responsible for the growth hormone taken from the rabbit or human being. Now, it is attached to the promoter. PR means promoter region of mice. Now, as we observe, this is the gene responsible for the growth hormone. This is promoter region derived from the mice. Now, it is attached to a vector. Vector means carrier. Here, what is the popular vector? PBR 322. Here, PBR means plasmid, Bolivar, Roderick. Bolivar, one scientist name, Roderick, another scientist name. These two persons popularized plasmid as vector. Its number is 322. Generally, the plasmid 322 vector is used for the experiments. Now here, transgenic animals can be produced with the help of what type of gene? PBR 322, not 372. 322. Now the answer is